Pretty thighs, shawty, she a dime. Demon girl, evil eyes, she be telling lies. Slim waist,
Today, it is my pleasure to interview Dr. Robert Tjian, the former president of the Howard Hughes Medical Institute and co-founder of Tularik. So, Dr. Tjian, thank you for being part of this episode. With such a illustrious career, can we start with you taking us back and telling us about your background?
Well, actually, let's see. I was born in Hong Kong, like you,
Yes.
but I didn't stay there very long. I think I left when I was about 3 months old and traveled to South America. I lived in Argentina and then Brazil, and then came to the United States when I was 13 years old to go to high school. After that, I went to Berkeley as an undergrad and really fell in love with science there. I had started off as a math major, but I quickly realized that I was really not that good in math, but that biology and chemistry were really something that I could relate to. So that's how I got my start. And I worked with a very famous biochemist by the name of Dan Koshland, who was really an enzymologist. And I learned both biochemistry and chemistry with him. And then I spent a year in Oxford, England, doing X-ray crystallography with Sir David Phillips. So that was a fantastic experience before I went to do my PhD at Harvard. Even though everybody thought I would go there to continue my crystallographic studies, I really decided I wanted to switch. One of the things that I have done throughout my career is to like switch fields or switch systems about every 5 or 10 years. And I think that's really kept me going. It still does. But there at Harvard, I accidentally met a young scientist at the time, just starting up his lab, Richard Losick, who introduced me to the whole concept of gene regulation and transcription. That was in 1972, believe it or not. And I'm pretty much still completely absorbed about trying to understand gene regulation and transcription particularly to this day. And it looks like we still don't really understand what's going on, especially in humans. So after doing my PhD on bacteria gene regulation, bacterial sporulation, and a phage virus, SPO1, I really learned about the intricacies of gene regulation from studying that system at the time. And this is before, really the major revolution in recombinant DNA and, that whole period, I know I listened to your interview of Tom Maniatis. We were contemporaries at the Harvard Biolabs. We both ended up at Cold Spring Harbor. So I went to Cold Spring Harbor. Really, I was recruited by Jim Watson, who was a Harvard senior fellow while I was a Harvard junior fellow. And he really wanted me to work on a protein called the SV40 tumor antigen. He had an intuition that was going to be an important protein, more in the sense of how the virus replicates and causes tumors. But I was really interested in it because it was also a transcriptional regulator. And that's really where I got my first start at truly working on eukaryotic transcription as opposed to prokaryotic transcription, which I did at Harvard. And really that launched my career. Being able to do biochemistry on the SV40 tumor antigen allowed me to purify basically the first DNA binding protein from a eukaryote. And from then on, it was just trying to understand how transcription really works in animal cells, in human cells, and ultimately leading me to trying to understand how that could help us think about disease.
Yeah.
So that's sort of my career.
Yeah, I think you mentioned by now that you're moving around different places. So I kind of had the same experience. I lived in 3 different places right now. So I was like, what was it like moving around, just like embracing and just adapting to different countries, like cultures?
I thought it was fantastic. I mean, my family moved a lot during the early days. And all my siblings, I had 8 siblings, or had 8 siblings. Some of them, unfortunately, have passed. We were spread all over the world. So we never felt uncomfortable anywhere. Very adaptable family. We learned a lot of languages. At one time, I spoke 4 languages fluently. And so I think that moving around makes you, appreciate differences and similarities, but also makes you really understand different cultures. And I think all of that comes in later on in your life and opens you up to other ideas. And my children are likewise, they love traveling and they love embracing other cultures.
Yeah, I kind of have this similar feeling, type of like, I can't, it's like adapting different cultures just makes me have like the wider perspective on things. That's in my opinion.
Absolutely. I think it may be comfortable to stay in one place your whole life, but it's quite limiting.
Mm-hmm.
And so I think, and my wife is from her family's from France, and so we have really broad interests. So, and I think that also helps in terms of as a professor in training people and understanding the different cultures and how people are motivated differently. So I think all that comes into play later on.
Mm-hmm. Yeah, I mean, you were a postdoc fellow at Dr. Watson's lab. So I meant like I know you have a lab right now, but like what did you take away from his lab that you kind of incorporated into your lab?
Well, it wasn't just Jim Watson's lab that I did that. I absorbed a lot about the way Dan Koshland ran his lab in Berkeley. And then, of course, in my years at Harvard with Rich Losick, which, by the way, I just celebrated his 80th birthday a couple of weeks ago at Harvard. I took away a lot of lessons that Rich, how he ran his lab, and then, of course, Jim. And all 3 of them are very different, but all had fantastic ways of doing things, which I incorporated into my own way of teaching people and running labs and asking questions that sort of fit my own personality. So I think you take bits and pieces from each one of them. The other thing is that I remained very close friends and colleagues with each of them throughout their lives, which to me was also very important, that they're sort of like part of my, my family, really, my scientific family.
Mm-hmm. Yeah. So why did you? So I know you're in the Boston area in your PhD. So why did you want to go back to the West Coast? Because I know all the scientific research and biotech, they're all concentrated in Boston. I mean, especially during your time.
Well, that's a really great question. I think most people went, especially because I was a Harvard Junior Fellow, I probably could have gotten a number of very nice positions at MIT or Harvard. And I absolutely did not want to do that. I really felt that my personality fit better the West Coast style. Harvard and the whole East Coast, has more of a sort of top-down sort of tradition, whereas the West Coast is sort of, you're a young professor, you get to do what you really want to do, and no one's going to tell you otherwise.
So you're saying it's like the Wild West kind of in the West, in the East, it's kind of more structured.
Well, there's more structure and sort of, a little bit more top-down sort of influence, whereas in the West Coast, and I really felt that, particularly at Berkeley, where, I was there during the free speech area and a lot of rebellious nature, which I loved. I really wanted students that had that sense. And to this day, I would say that is what defines the true differences between the types of students I get at Berkeley versus the ones that I would get probably at Harvard. It's not that they're any smarter. They have a different motivation.
I see.
And to me, that was really important, that I was going into new areas where I didn't really want the big, well-established senior faculty to tell me what to do. I wanted to fail on my own if I was going to fail, or succeed on my own. And that's exactly what it was easier to do that in Berkeley. I'm not saying that you couldn't do it at Harvard, but it was harder.
That's it. Yeah, I mean, no, I know that during Dr. Ross, during your time at Cold Spring Harbor, you found the first protein of virus which can regulate the gene expression in mammalian cells, is the SV40 large T antigen. So, no, can you explain why this discovery is so important and what fields did this protein unlock?
Well, back then, nobody really understood exactly how transcription was going to work. And I would maintain that today we still don't completely know, but we know a lot more than we did then. But at the time, there were certain models about gene regulation that were really based on what we understood about bacteriophages and bacteria. And some of that, was able to be translated into the eukaryotic system. But the more we studied the eukaryotic systems, particularly the mammalian systems, the more we realized that things are quite different. And so back then, just getting your hands on a protein that could actually recognize DNA sequences quite selectively was a huge technical chore, because these proteins are usually produced in very small quantities, and you had to really be a very good protein biochemist to be able to purify this. And the way we purified it back then was very different from the way people purify now, because you can clone anything you want, right? Just put a tag on it and you can use an antibody and pull it out. Back then, you had to do it the hard way, which is there's no tags, there's no molecular cloning. You had to figure out how to purify the protein. So it was a technical challenge to do that. And then to develop, entirely new ways of measuring DNA binding, how is it specific? And, of course, it unraveled and discovered an entire world of DNA binding transcription factors, as you probably know, and that pretty much changed the field. So I think that first protein, the first one's always the hardest. But it's as it turns out, the first thing I did when I got to Berkeley was to purify a true human protein. That SV40 is a viral protein. So you might say, well, that's easier. And it probably was easier. So the next challenge was, could you do it in actually a human protein? Mm-hmm. And that was SP1. And that probably was even more difficult, especially ultimately cloning it, sequencing it, determining, its structure function relationships. And to this day, there's still graduate students in my lab studying how this protein works, because, it's a big protein with some really crazy structures and areas that are completely unstructured. And so really trying to understand the whole intrinsically disordered regions of proteins is a fascinating, challenging area right now.
Mm-hmm. Yeah, so I have a question, and I know you mentioned like back then, during your time, it was more difficult to do certain things. So I think I remember scientists saying that like scientists right now in the modern world can't survive in the 50s and 60s and 70s during the scientific era. Do you agree with the statement?
That they couldn't survive? What do you mean?
It's like they couldn't really do as much research as back then, because of the fact that the methods back then were a bit, again, like tough and hard.
No, I don't subscribe to the idea that the scientists today are any less talented or capable than we were back then. I think there were fewer of us. The technology was much more limited. But I think that the really talented early stage career scientists, students, postdocs today are just as capable, and in some ways more capable, because they not only have to understand biochemistry, molecular biology, and chemistry, they also have to be computational experts. They have to be essentially engineers. So in many ways, I think it's even more challenging. It's just different, So I think truly motivated, talented people, regardless of era, you have to overcome major barriers, technical as well as conceptual ones. And in many ways, things were just a lot simpler back then. You didn't have a lot of choices. You didn't have to, learn 20 different technologies to do anything. Today, my students, whom I feel are exceptional, probably I would consider many of my graduate students now way smarter than I am. Doesn't necessarily mean they're going to achieve the same level of productivity, but they are intrinsically really, really smart. And more capable than anything I've ever seen. And so I don't feel like there's any big argument about the past versus the future.
See. So, you showed that the transcription factors also exist in higher organisms, not lower, not in a higher organism. So, I guess for the people listening who don't know what a transcription factor is, can you explain what it is?
Yeah, well, it's a little bit, I'll have to kind of make it simple. Think of a transcription factor as any number of proteins, and there's probably about several thousand of them in a human, whose job is really to sort of interpret the genetic information of your DNA. These are the proteins and enzymes, because some of them are enzymes that have to synthesize RNA from a DNA template. You think of them as like reading the blueprint of life. What makes us, each cell in our body, do what it does is whatever genes are being read or expressed, as we say. Mm-hmm. And the proteins that determine which genes are being expressed are called transcription factors, because these are the proteins that can read certain sequences in the DNA and determine whether that gene should be on or off, or very highly on, or only a little bit on. And this is where it gets really interesting, why I got into the biotech business, because it became pretty clear in the 80s that, many, many diseases, particularly diseases like cancer or metabolic diseases and others, it's not that you necessarily have a mutant that's being the problem, but sometimes the problem is you're just making too much of something, or you're making not enough of something, or you're making it at the wrong time. Those are fundamentally gene expression problems. So some gene regulatory event, some transcriptional event has gone a little bit bad. And now you're either making too much of the oncogene or not making enough of the tumor suppressor, or, whatever it is. And so that's really what allowed me to jump from thinking about very fundamental gene regulation mechanisms to something like Tularik, where we're basically trying to manipulate that process and try to control it in certain diseases.
Mm-hmm. So yeah, I kind of talk about Tularik. I know when Dr. David Godell asked you to co-found Tularik, you said you didn't want to, but then you kind of changed your mind. So can you tell me why you said no initially, and what kind of made you say yes? I'm going to become and join the biotech world.
Well, it's not that I said no. It's like I didn't want, Dave Godell was kind of like the founding scientist, really, of Genentech. You're not going to, it's really hard to do another Genentech, right? Mm-hmm. So he had tremendous success, and he was really responsible for, the first 5 products out of Genentech. So my view was, you can't beat Genentech at its own game. You're not going to do another Genentech. So it has to be something different. So when I've discussed this with Dave early on, I really liked the idea of starting something with him, because I knew how talented he was, but I wanted to do something different. Now, Genentech and all of the biotechs at the time, Amygen, Chiron, they were mainly about biological products, right? They found a protein like insulin, a growth hormone. They figured out how to overexpress it, and they made large amounts of it, put in the bottle, and sold it. And so that's still a really important model today, but I was interested in trying to change the way we thought about it. And I thought, okay, for gene regulation, you probably need a small molecule that hopefully could be oral bioavailable, or could be an injectable, but ideally an oral bioavailable one that could actually manipulate gene expression through its interactions with transcription factors and the core transcriptional machinery. So that was really the concept. And in talking to Dave, I think he understood and appreciated the fact that this was a really different way of thinking about doing a biotech. to really go after high throughput screens using small chemical compounds. That was not what the biotech world was about. So we were stepping into unknown territory there. And this was before, companies like Gilead really switched over to study HIV and again, using small molecules. So I think there was kind of an inflection point where biotechs went from doing purely biologics to realizing that, and Tularik, I would say, was probably the first with not just Dave Goodell, with Steve McKnight. The three of us really felt that we could use modern gene regulatory molecular genetics and molecular biology to do drug discovery in a very different way, developing both biochemical assays and cell-based assays. Those were the two super powerful ones. And in those days, almost nobody really did robotic screens, high throughput screens, which is, completely conventional today. So to me, it's not that I wasn't interested. It's just that I felt like I needed to have a clear idea of how was Tularik going to be differentiated from other biotechs before I wanted to get into it. But once the three of us saw that this was really a possibility, we went all in, and obviously we really enjoyed this very fruitful collaboration and relationship with the three of us for over a decade, which is very unusual for founders. So to me, it was a fantastic, positive experience. And I learned a huge amount about drug discovery and about organizations and about leadership and so many things that later on, helped my career.
Mm-hmm. Yeah, I mean, you mentioned Tularik was one of the hottest biotech companies in the 90s. So did you ever envision it becoming being on the stock market?
Well, I mean, you don't start a biotech company of any kind without seeing some sense of there has to be some event for liquidity. In our case, we did both. We went public and then was acquired by Amygen. Both things were very, very positive. It really allowed us to develop drugs in a more sophisticated way. Small biotech, especially back then, but even true today, we're usually really, really good at doing the drug discovery side, but we're not usually very good at the development side, the clinical side. So, without Amgen's very, very sophisticated clinical group, we would never, be able to advance any of our products. So to me, that was a huge lesson. And that's, playing itself out again in one of the more recent companies that I started, Icon Therapeutics. So it's, I think it's still the fact.
Mm-hmm. Yeah. So did Tularik ever commercialize any products or have any patents?
Oh, I had lots of patents, and it took several products into the clinic, but I don't believe any of them ultimately made it to the bedside.
Mm-hmm.
And that's not unusual, as you know. The probabilities of it making it to the bedside are very rare. Mm-hmm. And some of it was not because it wasn't effective. You have to take into account all kinds of reasons. Whether it was financially viable to do that, was patient population big enough, et cetera, et cetera. So I don't believe any of our products ended up without much, much more development from Amgen getting anywhere.
Mm-hmm. So I think I know Tularik was losing money every year. So do you estimate how much Tularik was losing each year?
No, I don't remember. Every biotech loses money. The more important question is, can you keep raising money, which we were able to do to keep it going? Because let's face it, it takes maybe a decade before product is going to be actually going to be making you any money. So that's one of the biggest challenges of a biotech, is how do you sustain, even if your science and your preclinical work and even clinical work is going well, you still have a long path to go. So, I can't think of too many biotechs that are making money, until much, much later, and usually after some acquisition event, or if they get really lucky, a product that actually makes it through.
Mm-hmm. I know Tularik was having a negative cash flow. So why do you think Amgen just wanted to acquire for 1 billion or maybe more than
Yeah,
that?
well, this is the one of the most interesting things that I learned about is that, if you look at a company like Tularik, there's really two big assets that it has. One, of course, is the products themselves, which, have promise, but there's no guarantee that it's going to make it all the way to the end. But there's another product that I think is perhaps more important, which are the people that's there. It's kind of like you're acquiring an entire team. And I think that is where Amgen's leadership understood that we had really, really high quality research development teams. And to them, that was perfectly reasonable for them to pay what they did, over $1 billion for that team, which, to this day is still, Amgen North. So I think that it's not just about the product. In many ways, you can think about the people as being more valuable because they're going to generate many, many products over time. So does that make sense to you?
Yeah, I see. So it was more of the talent that I think Amgen wanted, not like the products or the
That
yeah.
that certainly one. I mean, it's not that they didn't think the products were worth looking at, but I would say it was smart of them to realize that it was also the people.
I see.
And
So,
I would argue that the same thing was true maybe for the relationship between Roche and Genentech. Yeah, but Genentech had products, but actually perhaps the more valuable was the teams that they had built.
I see. So, what is the difference between like between managing a lab and managing scientists at a biotech company?
Well, at one level, there's not a whole lot of difference because you're trying to do really, really good science. At another level, it's different because at the university and in my lab, I have graduate students and postdocs. I'm training them to be independent scientists. So they're working on, especially the way my lab works, is that I don't tell them what to work on. I don't tell them what projects to work on. But graduate students and postdocs come to my lab. They have a clear idea, usually, of what they want to do. Even the graduate students ultimately might spend the first year or two trying to figure out what it is that they want to work on. So that's very different from a company where, you don't have that freedom to say, oh, yeah, I'm going to go work on something that's, unrelated to what you're doing. So that's different. But fundamentally, it's all about doing really high quality, rigorous science, making sure that you're pulling out the truth, which in biology is very difficult. The other thing, of course, in industry, things go just a heck of a lot faster because you have so much, better resources and more people working on a project. You have big teams of real great experts. So, watching what's going on at Tularik and now at Icon, things go so much faster with so much more depth. So the industrialization of certain technologies that are sort of boutique in the laboratory and in an academic laboratory become really robust and, I would say industrialized in ways that in an academic setting would be nearly impossible to do. So I think they really complement each other in a most important way if we're going to tackle difficult problems.
Yeah, talking about your lab. I think your lab at Berkeley first also identified the first gene-specific transcription factor in the human cells was SP1. So why is SP1 so important in our cells?
Well, SP1 just happened to be the first. Don't think anybody would want to argue it's the most important. It is critical. Obviously, if you knock it out, you're dead. But, transcription factors in general I think of them as essential proteins that are also really dangerous. Because if you have too much of something or too little of it, you're in big trouble. And SP1 falls into that category. Now, in humans, there's, SP1, SP2, SP3, SP4. So there's been duplication of that gene because it is playing important roles in various different biological settings. But it's important from the discovery standpoint, because it was the first truly human protein. And its structure function revealed some still mysterious aspects. It has a zinc finger DNA binding domain, but then it has a lot of so-called intrinsically disordered regions, which to this day, we still don't fully understand exactly how it all works. In fact, I have graduate students in the lab right now trying to figure that out.
Mm.
And so this is why I find so fascinating about transcription factors in general is that, I've been studying them for over 45 years and I still don't fully understand how it works. There are not too many problems you can work for that long with as many resources as I've had and still be fascinated by it. So to me, that's like, I just got lucky.
Why do you switch some of your research focus from like in vitro biochemistry to approaches to
to the in vivo system? Yeah.
Yeah.
So that's a really great question. So as you are well aware, back then we didn't have any choice. So there was sort of like two major approaches to trying to understand complex mechanisms like transcription. Mm-hmm. One was to use, classical genetics, make mutations, see what happens if you make a mutant in a particular protein like a transcription factor. That's incredibly powerful, and especially if you're working in systems like Drosophila, the fruit fly, or in yeast, where you can manipulate the system really well. The complementary approach is in vitro biochemistry, is sort of the opposite. You take the cells that you're interested in our case, the famous HeLa cells, which is in the news right now, and you grind it up, you purify the components that you care about, and you reconstitute the reaction that you think is important. In our case, messenger RNA synthesis with a specialized template. Well, that's beautiful. So it allowed us over the last 20, 30 years to sort of, at the very least, kind of have a shopping list of all the proteins that are necessary for the process to happen. So it's really good at identifying the players. It's less good at trying to figure out how the players all interact with each other in a natural environment. So, because in a test tube, you're purifying things. First of all, the concentrations are very different than what it is in a living cell. And second of all, when that same transcription factor in a living cell is in the presence of, 1,000 other proteins that are not in your test tube. And if they were in your test tube because you're throwing it in there, they're not in the right concentrations. And so it's virtually impossible to fully recapitulate the reaction conditions of the in vivo live cell system with a test tube reaction. And, I've been aware of that for a decade or so, but it's kind of like, yeah, I know that, but what can I do about it? I don't have any way to get around it. Until, Eric Betsick and others developed super resolution microscopes that would actually allow me to watch individual proteins in live cells in real time doing their work. I did not think in my lifetime as a scientist I would be able to have that privilege. And to me, it was probably one of the most exciting and important things that I've ever done, is switch from doing classical reconstitution biochemistry to doing live cell imaging, which I call it in vivo biochemistry. Mm-hmm. I'm still asking the same mechanistic questions. I'm still trying to understand how complex reactions happen, but now I'm asking it in the context of an entire living cell, and for that matter, whole organisms or even whole organoids. Okay. Mm-hmm. That's a huge, huge step forward. And this is really why I started Icon Therapeutics, because I feel like, just like I felt our understanding of transcription factors back in the late 80s, early 90s allowed me to think about starting Tularik, this new insight into the molecular dynamics and kinetics of transcription factor function allows me to think about a new way to do drug discovery. And so Icon is basically that, is how do you use these microscopes to measure the movement of drug targets in real time in the presence or absence of drugs? That is incredibly powerful.
Man. I think from talking to you about your different companies, like you seem to be like kind of like ahead of other people by like a step forward. If you kind of know where I'm going towards this, like for example, for like all of us, right? Like each of your ideas, like you're always like ahead of like some people.
Well, I don't know that this happens in science all the time, but that is what I want to do, right? Yeah, I don't think it's fun to just do the same thing over and over again. As we talked about before, I'm always switching technologies. I'm always switching systems. I went from HeLa cells to Drosophila, from Drosophila to human embryonic stem cells. I mean, I don't think we should be limited in how we go about doing this. Now, I'm not smart enough to do all of that on my own. This is no way. I am so dependent on and excited about working with students for that reason, because they come in and it's like nothing phases them. I mean, this is what I love about Berkeley students is the harder the challenge, the more they're into it. And that's hard to find. And so the people in my lab are way more experts at doing some of the stuff that we're talking about than I would be at this point. I mean, I'm kind of the conductor, but I really don't know how to play the instruments individually anymore, right? So, but yeah, to me, starting a new company isn't just trying to make more money. That's not the primary motivation for me. Never has been, even with Tularik. It's more about, can I use my science in a way that might help advance not just my own work, but other people's work that will allow for discoveries that will make drug discovery and drug development more efficient. Because let's face it, the United States is still the primary country developing novel, unique drugs, and it is incredibly expensive and time consuming. And anything we can do to try to, make that more efficient, a little bit more rapid, it's going to have a huge impact. So that's what motivates me.
Yes. So, I know you have some stem cell research going on at your lab. So what do you think about the future of the whole stem cell tech?
Well, I think that, we live in a really exciting time because you not only have all the advances in stem cell technology, you have CRISPR-Cas9 gene editing, you've got imaging, you've got AI machine learning tools. I mean, I use all of it. I can't, there's not one thing that I feel is the only thing that's going to be the key. We use a lot of chemistry. We use a lot of, gene editing tools. I would say a company like Icon couldn't survive with all of that. It's a combination of that and robotics and, really, really sophisticated data analysis. So to me, being where I am here in the Bay Area, in Berkeley, is critical because I have to be surrounded by people who are experts in all of those things. And there are not too many places in the world where I can get all of that, And so to me, I'm super happy to be in a place where I can pretty much walk down the hall and get the world's expert on pretty much anything. And so it's that convergence of these different technologies that excites me the most. Yeah. And again, if you go read up on Icon, you'll see that it's really hard to imagine. Half the people there are engineers. They're not molecular biologists.
Oh, really? Wow.
You should go and read up on what Icon Therapeutics is about, and you'll see what I mean. That they're generating over a petabyte of information every other day on imaging technology. So big data science is what they're about. But at the same time, they're just as deep on the biology and the molecular biology and on the clinical side. I think it's a completely different model of what a biotech looks like. And so to me, that's exciting, right? I'm not just doing another cookie cutter biotech. It's a completely different thing. It's about a completely different platform that will empower drug discovery to be in a completely different place a few years from now. And to me, that's worth doing. Exactly how it's all going to work, I don't know, because you never know until you get there. But obviously, again, it's about people. It's about being able to recruit the very, very best. I mean, being able to get the president of Merck to become the CEO of Icon, that's huge. So
No,
I
yeah,
think it's all that. It's all these lessons that I learned from Tularik, but applied in a completely different new context, completely different technology, and different times. We're in 2023 here, and Tularik was 1992, so it's a long ways, but yet same lessons.
yeah. I mean, I know you joined the board of directors for Nurex. So what made you want to join this private com drug
Well,
company?
actually, I was one of the founding members of Nurex, but then because I went off shortly thereafter to be president of the Howard Hughes, I could not be involved anymore until many years later when I came back and stopped being president. That's when I joined the board again. And Nurex, again, it was one of the very, very first companies that did PROTACs, or what's called induced proximity. And, again, it was all about people. It was about putting together John Kurian and Misha Rappe and together to go after, ubiquitin biochemistry for drug discovery. And in fact, I just founded another company with Misha Rappe called Tjian that's going to do the same thing that Nurex did, but now apply not to oncology, but to neurodegeneration.
Mm.
So I'm always thinking about how to be differentiated, but at the same time have enough experience to know, has some probability of success. As you know, drug discovery is incredibly difficult. Most of the times we fail.
Mm.
I'm always trying to figure out how can we increase our probabilities of success. And that's the reason for doing ICON. It's the reason for doing Lyterium.
Yeah. I mean, I think you also joined the Colin Group as Life Science VC group. So as I guess, an investor or partner, like what types of elements do you look for in an entrepreneur when you invest in their startup?
Well, the Column Group was started really by, Dave Goodell, Peter Svenelson, myself, and a few others, almost like a reaction to after we did the mergers and acquisition with Amygen for Tularik. We learned so many lessons from doing Tularik that we felt that we could probably contribute to the venture capital world through our knowledge of how biotechs should operate. And certainly Goodell is unique in that, in his experience. And so again, I couldn't stay there because I had to go off to HHMI for 8 years. But when I came back, it was really stimulating to be part of that group. And all the things we've been talking about are things I'm looking for. I personally, I'm not saying all my partners at the Column Group feel exactly the same, but we kind of want to do things that are differentiated, that's not just, the same as every other biotech strategy. There has to be something unique. It's either the people or the technology or a combination of the two.
Mm.
And there's no set answer. And being a venture capitalist is very difficult, and certainly a lot of it has to do with luck, as well as the ability to recruit the right people.
So it's kind of like Shark Tank, right? For the people out here listening.
Yeah.
Let's see. So has Colin ever invested in startups that have failed?
Oh yeah. And there's no way that you if you're in venture long enough that you're not going to fail. The question is, can you fail less frequently? So, yeah.
So no, in your opinion, like why do you think these startups failed?
Oh, for all kinds of reasons. Sometimes, the fundamental problem is we don't know enough about, in many, many cases, we don't know enough about the true molecular mechanism and the sort of pathophysiology of disease to know for sure exactly how a drug's going to behave. And not only that, in many, many cases, as you know, we're stuck doing the preclinical studies on animal models, which are not necessarily a very good reflection on what's going to happen in the human. So more often than not, it fails for any number of reasons, but primarily because, you can't actually test it in the system you ultimately want it to succeed. So that's a big problem. A lot of times the drug is hitting the right target, but it's pharmacokinetic and pharmacodynamics just doesn't work. And it doesn't get to the right place at the right time. And so your therapeutic window is terrible. And there's so many ways in which you can fail. Because, after all, the criterion for success is that you got to convince the FDA, which has very, very specific sort of criterion. Some of it makes a lot of sense, and some of it sometimes doesn't make a lot of sense, but you still have to go by that rule. So, there are not too many industries that have to go through that. And so not only do you have to succeed on the scientific and on the clinical side, but then you have to succeed on the regulatory side. That's 3 strikes against you.
See, so you collaborate with Dr. Dodona on the response to the COVID-19 pandemic. So my question is what did your lab contribute to the pandemic?
Well, that was a really interesting sort of side gig, if you like, because once COVID announcement hit California, those of us in the laboratory weren't allowed to work on anything except COVID if we wanted to come into the laboratory. And fortunately, my students and postdocs didn't want to just sit at home twiddling their thumbs. They wanted to go to work, even if it meant switching completely into COVID. And it was really a lot of fun because different students and postdocs had different ideas. One group decided they were going to try to develop, in the early days, the assays weren't very good. You might remember that, So we were trying to develop assays that were faster, PCR-based assays that were more accurate and cheaper. So we did that. One group did something really practical. They just went in and said, hey, there's not enough hand sanitizers and personal protection equipment. We're just going to make them and give it away. So they did that. Another group developed, antibody assays so that, anybody that got infected, we could test. Like I tested myself all the time. I know exactly when I was infected and when I wasn't, and I know exactly which strain infected me and so on and so forth. So to us, it was like a lot of interesting, important thing. One of the really cool things that one graduate student in the lab did was to really develop a very sensitive detection of COVID in wastewater, in sewage, which is probably one of the best indicators of even now. Like most people aren't doing PCR tests anymore. Nobody reports on home tests. But if you look at the wastewater, the sewage viral load, you can get a very good idea of when the population is getting more infection, which is happening right now. So we did all of those things. But it was, again, completely under the control of the students and the postdocs. I didn't tell them, any of them, what they should work on. These were all ideas that they developed and they published on it. And so that was actually kind of an interesting time because students didn't want to just sit around and do nothing. They really wanted to come in, and if they had to work on COVID, so be it. And they did.
See. So I assume you guys were wearing masks and hazmat suits.
Well, it depends on what we were doing. If we were working with the virus, you darn right. We were in, very, very restrictive conditions. But most of the times we didn't work with the virus. In fact, it was actually difficult to get human viral samples from the hospitals. We had to get very special permission to do that. And we, of course, had to do it under extremely contained situations.
Yeah, so you were the president of Howard Hughes Medical Institute. So why were you forced to give up your positions? I know you mentioned that several times.
It's not a question of being forced. I was happy. I wanted to do that job really no more than 5 years. I ended up doing it 8 years. I really wanted to get back to my research, and particularly because I wasn't, as president of a nonprofit, you really can't mix things with for-profit. So I wasn't able to do any biotechs. As I told you, I had to leave the board of the Nurex. I couldn't do a column group. I couldn't do any of that. So, which was fine. I mean, I think that Howard Hughes is one of the most amazing nonprofit supporters of biology. And having been a Hughes investigator for so many decades before, I felt the responsibility. If they really wanted me to help them as president, I would be doing that. But I really felt like the sooner I could get back to doing my research full time, and to do my biotech stuff, the better. So it was a very mutual decision, I would say.
I see. So, I think during your time at the Howard Hughes, you did build experiments to communicate scientific research in new ways. So, what do you think are some ways to improve the scientific communication?
Well, I think, while I was president, I did continue my research. I do believe that scientific communication is a serious issue.
Mm.
And I think COVID made it even more acute because there's so much misinformation going on. It's like science is hardcore. There's no you can't make up stuff. It's people have to understand that there's truth and there's falsehood, and there's not, you can't mix those. So I think that scientists are not necessarily really good at communicating. And I think that's something that we're going to have to do better. And it's only getting worse because incorrect information can get out so fast that, and people will believe all kinds of things online that, for a scientist who knows what's going on, it's just crazy.
So, you were also the scientific advisor to the Chan Zuckerberg BioHub and Biomedical Sciences. So, what makes the Chan Zuckerberg Hub so different from the Howard Hughes?
Well, I think first of all, it's fantastic to have people like Mark and Priscilla even think about wanting to support basic science that will lead to translational science, ultimately to the bedside. And their goal of trying to, tackle major diseases over the next 50, 100 years is just a fantastic goal. But they're doing it in a very different way than HHMI. And, I had long conversations with Mark and Priscilla about what are the pluses and minuses of doing it this way versus that way. And they're doing it in a different, but I think equally powerful way. And they have resources to truly advance, particularly on the technological side and on the platform side, the science in ways that almost nobody else is doing. So I think we need many different models of how to support young scientists in their work. And I think theirs is a great one. I think the Hughes is another great one. But, as much as these are major efforts, they still pale in comparison to the amount of money being spent by the NIH. So we need all of it, and we need more. So I think that, as more and more high net wealth individuals realize that there are some major problems that are science related, whether it's health or environment, the fact that they're getting into that is really a positive sign.
Mm-hmm. So, I actually read on our website, you're actually a candidate for the 2014 Nobel Prize in medicine. So, how do you feel about getting snubbed from winning the prize?
Oh, we get snubbed all the time. I think we get real used to it. And the truth of the matter is, I don't think I think much about it. I think it's those kinds of awards are wonderful, but I think they're just as political as anything else. And so it's not just about the science. And so once you have a human element involved, then, all bets are off. So I don't think one can really worry about that. It's a great honor to even be thought of as being a viable candidate, which I think, that can't be very motivating for me at this point.
I see. So what advice did you receive during your career that shaped your professional development or success?
I don't know if it was advice, but just a realization that you got to do what you really care about. At my stage at my age and at the stage of my career, why am I still so enthusiastic about transcription? It's because this is what I really love doing. This Sunday morning, why am I in the lab? Because this is what I love doing. I'm going to go, as soon as we get done, I'm going to go I haven't talked to my students about, what their experiment last night was about. So you got to do what you love. So it's not a job. It's what I would do if I even wasn't getting paid for it. In fact, I'm not getting paid much for doing that. So to me, I think that's what's important is, what do you want to get up in the morning and think about? So to me, it's kind of, in one sense, should be obvious, but on the other hand, I feel very privileged that I can do that. Not everybody can do that. Mm-hmm. So to me, getting in that position where I can actually spend all day long thinking about what I really care about and interacting with people I really like interacting with, smart students who are giving me new ideas, and how much better can that be?
So is there anything else that we didn't talk about that you want to talk about right now?
I don't think so. You covered a lot of ground.
All right. Thank you, Dr. Tjian, for being part of this episode and giving me the opportunity to interview you so people can get to know more about your insights and your transcription factors in the biotech business.
All right, Karen. Thanks a lot. Good luck.